It's my great pleasure tonight to introduce our guest lecturer, Dr. Stevenson, a research professor at the University of Arkansas and one of the world's leading experts on slime molds. I had the very good fortune of attending Dr. Stevenson's course on the same subject at the Eagle Hill Institute earlier in the year, and I've just been completely fascinated by these incredible organisms ever since. And I just know after this lecture, you will be too. Dr. Stevenson has literally written a book on the science of slime, as well as several other mycology books. And whether you know a lot about slime molds or know nothing, make yourself comfortable for a fun ride tonight. Slime molds are just the coolest. So please join me in giving a very warm welcome to Dr. Stevenson.
Well, thank you, if you could hear me. Okay, an introduction to Mixomycetes. There are slime molds other than Mixomycetes, but for the average person, a Mixomycete is a slime mold is a Mixomycete. And the first thing I would say is that even though they're small and usually rather inconspicuous, if you keep your eyes open, especially if you're a mycologist looking for fungi, you're going to spot them. Now look, gee, that's the end of a log that's decaying, and I wonder what those red things are. Well, I know what they are. You can probably guess. Whoops. Now, I've gotten lots of questions over the years, and also questions as of my sanity and why I study them. But again, they've been very good to me, and they've allowed me to go all over the world, so I can't complain. But what the heck are Mixomycetes? Well, they're fungus-like, at least somewhat. Now, for a long time, they were thought to be fungi, and even people like Alexopoulos argued that they were fungi, but he was wrong. The molecular biologists indicate that they're amoebozoans. They're actually a lot more closely related to, say, an amoeba than a fungus. There are only about 1,000 species worldwide. So, and I know every one of them. No, I don't. No one does. It's not something the average person knows very much about. That's certainly true. Most people do not even know that this group exists, which is unfortunate. Most are small and easily overlooked. I've indicated that. And the last thing, it's really depressing. Being called a slime mold is, well, that's depressing. It's not a particularly attractive common name. There's got to be a better name, but well, they don't have it. As I just referred to, if you look at the relationships of the group, well, they're not related to that nice Amanita, but they are closely related to that amoeba. So something that, again, we've become increasingly aware of. Why study them? Well, there are places in the world where they've never been studied. That's a nice incentive. We know relatively little about their patterns of distribution worldwide. I'm talking about boreal zone, temperate zone, tropical zone. We don't know a lot about their role in nature. As we learn more, we discover that maybe we have underestimated their importance. Now, how they interact with other organisms, it's still largely a black box. Something goes in, something comes out, but we don't know what happens inside the box. Probably the biggest splash of publicity the slime molds got was a lead story, the cover story, in the science section of the New York Times in 2011. Wow. And they used these images. I supplied those images. And someone just casually looking through the paper that day, I think they'd be drawn to that. They said, whoa, what are these? They look neat. And they'd probably read the article. I will say that when that article appeared, I heard from former students that I hadn't been in contact with for 30 years. They read the New York Times. Wow. Well, this is something that goes on in nature that, again, we just don't have a lot of answers, but slime molds are not toxic. They're not poisonous at all. When the fruiting bodies form, they certainly can't move. Well, as I tell my classes, it's just like an M&M. Most of you know what M&Ms are. Well, you could go through the woods and just broadcasting M&Ms, just scattering them everywhere. And then the next day, I shouldn't have done that. I'll go back and pick them up. I don't think you're going to find many of them. They're good to eat. Well, slime molds apparently are good to eat too. And this little fella is munching away on them. Now, there are two ways of studying slime molds. And again, for us, for me, for this presentation, slime mold, Mixomycetes, they're synonymous terms. I may switch back and forth, but no big deal. Now, you can go out and collect specimens that have developed under natural conditions in the field. And that last presentation, we saw several slime molds. Wow. Now, but it's also possible to grow your own. Bring in some organic material, put it in some container that holds moisture. We call it a moist chamber culture. Europeans usually refer to as a humid chamber, but we use petri dishes, which you'll see, but anything that will hold moisture, you can actually put the stuff in there, add a little bit of water, usually pour the water off, most of the water off. Watch it for a few weeks, and it's crazy how many slime molds you'll get. In fact, it's not unusual for some substrates, good types of bark, for example, to get 6 or 7 species in one petri dish, 6 or 7 different species. This is a field collection, or it's not collected yet, but a specimen of fruit in a field. Very, very common, especially late summer, actually starting this time of the year and going on until October. Trichia Veracosa, or Vesperium in some books, but it should be Vesperia. This is a moist chamber culture. Those are pieces of bark, a filter paper, which retains some moisture. You, again, put the pieces of bark in, cover them about halfway with water, usually pour the water off the next day or most of the water, put the lid on and watch it, check it. Ooh, this one fruited on the plastic lid of the petri dish, but it's a rather pretty one. Anyone who says, oh, that's not pretty, it is. This is a Diachia leucopodia, white-footed. The stalk is white, very aptly named, neat little critter. Now, some basic definitions. Now, some of these, certainly most mycologists would know, maybe all mycologists, lay people not necessarily. A habitat, a particular type of ecological community, a forest, a grassland, or a desert. Now, obviously, there can be different kinds of forests, a coniferous forest, a deciduous forest, whatever else, but you're talking about a bigger thing. What is a microhabitat? Well, it's one of the component parts of that larger habitat. Micro means small. So you walk into a forest, the trees have bark, and actually bark is a good substrate for Mixomycetes. That's one microhabitat. You look down and you're walking across wood litter with dead leaves on the forest floor. Well, that's another microhabitat. You stumble over a decaying log. Oh, that's another kind of microhabitat, and so on and on. I'll come back to that, but habitat the big, microhabitat the small. Now, what are the microhabitats that could be examined for Mixomycetes? And you say, whoa, that's quite a lot of those. Not all of these occur in temperate forests. Many of them do, but some don't. Coarse woody debris, that's that log or stump. Ground litter, those dead leaves I just referred to. Bark surface of living trees, well, I mentioned that too. Wait a minute, dung of herbivores? Well, wait a minute. An herbivore eats plants, right? Well, the dung of an herbivore is processed plant material. Oh, Mixomycetes slime molds occur in association with plant material. There are actually species that occur, so far as we know, only on dung. Wow. Aerial litter. Not a lot of aerial litter in temperate forests, but in tropical forests, it's all over the place. Aerial litter is simply dead plant parts that are still attached above the ground. They've never fallen to the ground, never been in contact with the ground. Aerial litter. Lianas. Well, you say, well, we have grapevines, don't we? Well, we do, but we don't have a lot of lianas. Go to a tropical forest and you're blown away. Inflorescences of large herbaceous plants, typically tropical plants. For those of you that know the bird of paradise, you can grow that here, but you go to the tropics, go to Costa Rica, the heliconias and things like that. Well, I'll show you. They're wonderful habitats for mixos. Epiphyls. You know, we don't really have this here. An epiphyll is a plant that grows on a leaf. Wow. Bryophytes. Well, we certainly have bryophytes here. There are some Mixomycetes that seem to be pretty much faithfully restricted to bryophytes. Soil. Whoa, this is one we know the least about. Mixomycetes, at least one stage in the life cycle, abundant in the soil, and probably, well, there's no probably to it. We obtained enough data to know that they actually have a major ecological role, especially in what is called the microbial loop. And it's not going too far to say that if, for example, I had a magic wand, which I do not, and I said, bing, I'm going to get rid of all Mixomycetes. Well, I don't think I want to do that. But if I did, that would include all the ones in the soil. And I tell my students, the earth would stagger. Geez, wow. Well, they're doing a lot more than you realize. Submerged plant debris. This is something that wasn't realized for a long time, but if you go to a pond or a stream and twigs and leaves that have been on the bottom for a while, underwater, you can actually pull those out. Typically, let them dry out before you put them in the moist chamber cultures, but you get mixos from them. Succulent plants. Well, we're essentially talking about deserts. Yeah, there's some succulent, you actually grow ornamental cacti here, but, and actually, if you grow ornamental cacti here and you use moist chamber cultures, you occasionally get a species that's typically associated with a desert. Well, that's neither here nor there, but canopy soil. Well, I think I'll have to describe that when I get to the slide. We don't have that. Well, this is what Northern Virginia looks like, a typical forest. Anyone agree with me? Anyone agree with me? No, I don't think so. This is Western Australia. Those are Banksia trees. But if you look at it, bark of trees, you can see the litter on the forest floor, decaying logs. Well, the microhabitats are there, but it's certainly a different place in the world. This is a log, actually a West Virginia log, not too far away from you. The bark is off, and actually the best logs for Mixomycetes, the bark has, most of the bark has been shed. You have decorticated wood, as we call it. And in this case, it's covered with a layer of leafy liverworts. And that's actually a little community. There are all kinds of invertebrates in there. And actually, if you look very carefully, you'll actually find Mixomycetes that tend to be associated with that little community, that little microhabitat. Well, you know what this is. Those are dead leaves. Now, this shot was taken in Costa Rica, as I recall, but you look and say, well, these leaves are a little tougher than the ones we have here, unless you get into some of the oaks. But the leaves that you want tend to not to be the leaves that have just fallen to the ground. The one in the upper left, no, I wouldn't collect that. In fact, I'd probably remove the top layer and collect the ones that have decomposed a little bit more, take those back to the lab, and I'm going to have a pretty good chance of getting mixos. So what? Got to be a different community because the leaves are different. You're right. It is a different community, but those are leaves. Again, that's a leaf litter microhabitat or the ground litter microhabitat. Both terms are used. But there's something else I want to point out. That litter microhabitat is not homogeneous. There are different components. You see the arrows. You say, well, those are twigs, aren't they? Yep, those are twigs. Twigs actually support a different assemblage of Mixomycetes than do the dead leaves. Oh, you have to collect them, culture them separately. And there are actually some species you'd never find on dead leaves that you'd actually find on the twigs. This is actually leaf litter from northern Thailand. So it's certainly not northern Virginia, it's northern Thailand. That's the bark of a tree. You say, why? Recognize the bark. What you do to prepare that moist chamber culture is you use a screwdriver or a knife and remove portions of the dead outer bark. You don't get the inner bark. You don't disturb the tree or harm the tree in any way. But you saw that moist chamber culture earlier. The pieces that you get are postage stamp size or a little larger, and you collect those and take them back and moisten them. And again, it's rare that you make a moist chamber culture of bark and don't get some mixos. And you say, well, whoa, this looks a little different. You're right. There are epiphytes on that bark. Some lichens, a lot of bryophytes. There's some Mixomycetes you don't find on bare bark. You find them on bark that has a load of epiphytes. And we don't know why, but this bark, some of it, I'd certainly look at that. I think it'd be more productive than that bare bark, but that's based on hundreds and hundreds of moist chamber cultures. Now, not all trees were created equal. You know that. You say, whoa, that one in the middle, that has very smooth bark. Well, the bark would be tough to remove. It's very thin, but I wouldn't bother trying to collect bark to use for a moist chamber culture. It doesn't really attract, in other words, bark captures Myxomycetes spores from the air. Well, this doesn't capture very much. It's just too darn smooth. Now you look to the right, and there's a tree that looks a little more promising. Now it's true that in the tropics, there's a high percentage of trees with smooth bark. And so you look at, well, bark Myxomycetes or corticulous Myxomycetes, you don't remember that term. Maybe it's not the best place for them, but there's loads of aerial litter in the tropics, and wow, they're loaded Myxos. Well, some of you say, well, I know what that is. It's a vine, or a liana, if you maybe use a more technical term. Lianas have bark, just like trees, and some of them are amazingly productive for Myxomycetes. But if you really want to see lianas, this is actually the western part of Puerto Rico. It's literally sort of a vine forest. In fact, if you go to northern Queensland, Australia, there are forests that are actually named vine forests. I mean, vines everywhere. So you say, well, gee, that would be a good place to look for Myxos. It probably would be. Now, I mentioned the inflorescences of large herbaceous plants. That's a heliconia, actually in Costa Rica. And if you just walk by, say, oh, there's a bird dropping. Well, if you're a biologist, you say, well, maybe I better look at that bird dropping. And you take out your hand lens and say, oh, that's not a bird dropping at all. In fact, if there's a Myxo person with you and said, well, boy, you were wrong. That's Physmelium didermioides. Did you know that? And you say, well, no, I don't know Myxos. And the Myxo person said, go home. You're not supposed to be here. But I've done a lot of collecting in the tropics, and I've never collected that particular species on anything other than the inflorescences of heliconias. Interesting. I wonder what this is. You kind of have me buffaloed. Well, not really. That's dung. Now, my students, other people have joked. When I mentioned I study the Mixomycetes associated with dung, they asked me whether I follow, for example, a cow around with a paper bag and get the dung when it's absolutely 100% fresh. And I say, no, I don't. That's not the dung you want to collect. Go out in the field and find a cow patty, as it were. It works for buffalo too. And you actually find one that's gray and has a hardened surface, and you chip part of that off, and that's what you put in the moist chamber culture. In fact, I have to admit that last Friday in my lab, I was preparing moist chamber cultures with dung, cow dung. It kind of looked like that. The only thing is, it was cow dung from Queensland, Australia. Dung Myxos have hardly been studied in Australia, so there's a reason to do it. Ooh, that's an epiphyte. We say, well, that's got to be a tropical forest. Yes, in Costa Rica. But you see that arrow, that's aerial litter. Gee, that's dead material, but still attached. It's above the ground, and that's what you collect. And it seems to be unusually rich in Myxomycetes. In fact, in a wet tropical forest, the Myxomycetes seem to be displaced from ground litter to aerial litter because of the higher levels of moisture. Epiphyls. Well, most of the things you see on a leaf are little lichens, but they're the greener bryophytes. In fact, sometimes, and a person who's a specialist on this actually spied this sign, so that sometimes even on a leaf of that size, you can find 20 different species of lichens and bryophytes. That's incredible. But if you collect that and take it back and prepare moist chamber cultures, you'll get a few Myxomycetes. Now, typically their little fruiting bodies are little. They're abnormally small. And you typically in a fruiting get maybe one fruiting body, but they're there. Wow. They literally do occur everywhere. Well, those are mosses. There's some vascular plants in there too, but you say, what's special about that? Well, those bryophytes are actually on a projecting branch of a tree, well, in this case, in actually fjordland in New Zealand. That's neither here nor there, but this occurs other places. It occurs in any rainforest, temperate rainforest, tropical rainforest. But what if you see that mat of bryophytes with a few vascular plants, and you're curious, and you pull it aside. Look under the mat. You say, whoa, what's that? It looks like dirt, to use a non-technical term. It sure feels like dirt. And if you're really so inclined, ooh, it tastes like dirt. I don't know whether it tastes like dirt or not, but I certainly worked with it. Guess what? They're actually slime molds in that canopy soil. This particular shot was taken not too far above the ground, but I've collected samples from a canopy crane 40 meters above the ground. Yes, and the critters are there. Doesn't that look like soil? I mean, there are roots through it, but it's canopy soil, actually of a Nothophagus. You can see some of the leaves at the bottom. This is 40 meters above the forest floor in a tropical forest in northern Queensland. That's actually a fern, and what the person is doing, actually one of my colleagues, is removing some of the canopy soil that's accumulated at the base of the fern and putting it in that little plastic Ziploc bag and taking it back to the lab. He was actually working with cellular slime molds, dictyostelids, but we also looked at the Myxomycetes that were appearing in culture, and there are Myxomycetes there too. The interesting thing is, 2 of the species that we recorded in this study were new to science, and they're not known from the ground. And you say, well, how in the world? Well, how do they move around? I don't know. Again, one of those unanswered questions. Well, cacti. This is actually in Western Puerto Rico, where you actually get an interface between, well, broadleaf forest and a diffuse cactus forest. And you look at cacti and say, whoa, there are no Myxomycetes there. Well, yes, no. If a cactus has dead portions that fall to the ground, look at them. You say, whoa, there are Myxos all over the place. You're right. Some years ago, I was out west in Saguaro National Park, and where the prickly pear cacti and the pads fall to the ground, I just did an unofficial survey, I mean, for my own purposes. I just walked along, just a transect, turned over a pad. Are the Myxos there or not? Walk to the next pad, turn it over. Are the Myxos there or not? 7 out of 10 pads had Myxos present. Oh, that's a pretty high percentage. Now, if you're really thirsty, don't drink from that stream. But what's the guy doing? He's collecting submerged plant material. It's been in that stream for a while. Go back to the lab, culture it, see what he gets. Just an overview. It looks technical, it's really not. A, B, C, D. Well, you know your alphabet. What's A? Well, that's a fruiting, actually of a Stemonitis. What good are fruitings? Well, they're there to produce spores. What good are mushroom fruiting bodies? Same thing. They produce spores, lots and lots of spores. We think most of the time the spores are disseminated by wind, but not always. If a spore lands in a good place, it germinates. An amoeboid cell arises from that spore. In other words, a split or a pore develops. There are actually 2 things that can happen. That amoeboid cell, a Myxamoeba it's called, if there's water present, quickly develops flagella, you see on the right. In fact, we combine the 2 and just call them amoeboflagellate. whether they have amoeba, have flagella or not, amoeba flagellates. Now they actually act as little gametes, and if two of them like each other and they find them very compatible, they fuse. And that zygote is the first cell of the plasmodium, which is the structure that gives Mixomycetes the other name, plasmodial slime molds. That thing grows and grows and grows. It's really a single cell, a very large cell, and have many nuclei. The largest plasmodium I've ever seen in nature was about 3 meters across. And it was actually in West Virginia. It wasn't in some exotic place. But ultimately, if conditions are right, that plasmodium begins to transform from, well, basically gook, amorphous stuff, into some of the most intricate little fruiting bodies you could ever imagine. Wow. The thing that turned me on to Mixomycetes very early on was that transformation. I mean, you're going from a sow's ear to a silk purse and even more. I mean, wow, how does that occur? And it happens very quickly. Some Mixomycetes, that will happen in a few hours. Goodness gracious. That's a fruiting body. That's a pale, insignificant fruiting body, but actually one of the most common Mixomycetes in the world. Cinerea. Nice little stalk, has all the good parts, whatever the parts are. Well, fruiting body actually had parts that you can identify. They're fairly clearly discernible, as it were. At the base of a fruiting body, whether it's a stalk or not, stipend can be used to. There's a structure called a hypothallus, which is basically sort of residue left over when the plasmodium transformed into the fruiting body. Sometimes you don't see it. Sometimes it's very limited, but sometimes it's very extensive. And then there's a stipe or stalk, which elevates the spore mass above the substrate. Now, the actual spore mass is called a Sporathica. Now, a lot of books will just use something like sporangium, but Sporathica is the actual spore case. It's surrounded, at least in most Mixomycetes, by what we call a peridium. It's just a covering. Inside of that spore mass, in many Mixomycetes, but not all, there's a system of sterile elements called a capillicium. I'll show you examples and all that. And then the business part, there are the individual spores. Wow, the spore mass. So you look at that and you say, well, yeah, I think I could probably learn those. A friend of mine did this nice little illustration for me. He said, whoa, I think I can see A, that's a hypothallus. B, I wouldn't stump you on that. Stalk or stipe. C, that's the Sporathica. D is the peridium. Ooh. E is the capillicium. And F, the spores. Now you see that the stalk extends up into the spore mass. That's another structure. It's called a columella, but I didn't consider it important enough to say anything about it here. Now, spores. As was actually shown in that previous presentation, not all spores are alike. And for mushrooms, no way. Well, Mixomycete spores tend to be smallish, average about 10 microns. There are some that get up to push 20 microns, but the common ones don't get that large. But if you look at them, especially under a scanning electron microscope, some of the ornamentation is pretty exotic. The one on the left, that's warded, unless the warts are kind of sharpish and kind of spiny. But the one on the right, that's reticulate, but look at those reticulations. That's exotic. But those are spores. Those are spores of Fuligoseptica, which you've already seen, and they're germinating. Say, whoa, what happens then? Well, whoops, sorry about that. They develop in that. That's an amoebal flagellate. Well, that's a flagellum. There's really only one flagellum that you can see. There are actually two. And these things wiggle through the water and take in, they actually consume bacteria, fungal spores, other things too. But ultimately develops into this. That's a plasmodium. It's a neat little plasmodium. Again, the one I saw that was 3 meters across was actually a yellow one, not the same species, but they migrate I mean, if you were sitting on a log, you were 2 millimeters tall and you went to sleep, it would actually catch up to you and you'd find yourself inside a food vacuole, which probably is not a good place to be. So they do migrate more rapidly enough to capture and engulf some microscopic prey. Probably not a big factor, but they're capable of doing it. But ultimately, it transforms into a fruiting body. In this case, it's still transforming. And that's going to be Phyllogoseptica. Now, there are a few people in the world, you pretty much have to go to Mexico to find them, that actually collect it to this stage and fry it up and eat it. And supposedly it tastes like almonds, but I have not tried it and I don't think I will, but kind of interesting. I mean, they're not toxic, so you could eat them. Ooh, this guy is still developing. That's actually a little mixo called Phyllogoseptica, but you don't have to worry about that. But you can see that it's still gooey, slimy, whatever else. There are no spores yet. The spores haven't formed. Whoa, what's this? Well, I had well, actually, she was a postdoc from Vietnam who spent a year in my lab, and she was developing the techniques to grow large plasmodium. This is actually a plasmodium of Fizzerailies polycephalum, which some of you have heard about. We said, why was she doing that? Well, she was trying to develop enough so that she could demonstrate that you can actually use the plasmodium of Mixomycetes as a source for biofuel. Whoa. And she was able to produce a little biofuel. Now, what she's done since, which is kind of interesting too, the Mixomycetes produce all kinds of, well, molecular compounds. I mean, if you're a chemist, you can go into a lot of things. But what she's doing is looking at the products of Mixomycetes plasmodia, actually some other aspects of them too, and looking at possibly antibiotic properties, or even in some instances, anti-cancer properties. She's actually gotten some funding back home to work on things like that. Now, types of fruiting bodies, they're basically 4 types. Yeah, they integrate a little bit, but sporangium, plasmodiocarp, ethallium, pseudoethallium. You say, well, pseudo means it looks like something, maybe it's not, and you're right. Those are sporangia. They tend to be about the same size. Sometimes they're stalked and sometimes not. For this one, you can actually see the lower part of the protein persists. There's certainly a stipe or stalk. You can see the fuzzy mass is the capillicium, and there are loads of spores in there. Sometimes the stipe is longer than others. And so why is that? Well, we don't know, but if you go to the tropics, the same species occurs in Virginia and Maryland that occurs in the tropics. This is Trichia caliculatum. But if you measure the spore length, and we've actually done this, the stalk length, if I can say it correctly, the stalks, there's actually a significant difference. The stalks in the tropics are longer. You go, well, why? Well, we hypothesized in the tropics, and we're talking about the wet tropics, this guy really has to get the spores off the substrate and as dry as possible so they can be blown away. If they stay moist, they'll never get blown away. In fact, they'll be colonized by a filamentous fungus, and this fruiting, all of it will die. So how do you get around that? Well, produce a long stipe or stalk to elevate above that film of water, and seemingly Mother Nature has done that. This is a cute little guy. We're talking about something that's maybe 2 millimeters tall, incredibly intricate. So the Mixos don't have common names for the most part. I mean, Phyllogoseptica does, but I don't like to mention it. Dog vomit slime mold. Ooh, that sounds bad. This is sometimes called the Japanese lantern slime mold. I mean, it's incredibly intricate. This is, you saw that one developing. This is Trichia serpula. It looks like a serpent, a snake. Trichia serpula. This is actually on an old palm frond, actually in central Cuba. And that larger fruiting is certainly the size of my palm or a little bit more. Wow. If you were walking around, you'd spot that. This is the first Mixomycete discovered by mankind. Well, actually, that's not correct. if you had been born, let's say 500 years ago, which is possible, I mean, people, and you walk through the woods and you saw this on a log, would you notice it? How about that Fuligoseptica? Would you see it? Yes. The slime molds have been there all along, but it was in the mid-1600s before anyone described one. You know, this is a different organism. And the first one was Lycogula epidendrum, which is the one you're looking at here. That's an ethallium. What's a pseudoethallium? Well, ethallium is a large fruiting body. Well, pseudoethallia are large too, and they kind of, when you look at them, you say, well, wait a minute, aren't those sporangia? Well, in an evolutionary sense, they probably were sporangia, but they become all clustered or grouped together, forming a tight mass. So we call it a pseudoethallium. It's not quite an ethallium. These are the classic orders of Mixomycetes. Now, I'm going to have to say something. The classic orders. The Mixomycete classification systematics taxonomy has undergone some major recent changes. But the books that you're going to pick up, unless you pick up something really recent, this is what you're going to see. The Mixomycetes, the Kinosteliales, the Lyceales, the Trichiales, the Steminatales, and the Fizzerailies. And they kind of roll off your tongue. Mixomycetes, or the Mixomycetes. There's only one of them. Well, one of them you're going to find. There are a couple of species in the tropics, but they don't occur here. This is one of the species that occurs in the tropic. Spirosperma. Ooh, Morcella, after morels, literally, the genus. The Kinosteliales, the only one you're going to run into is a Kinostelium. It's a tiny little guy. I mean, you really need a microscope to see it, but this is probably the most abundant Mixomycete in the world. And if you prepare a bulk bark culture, almost always you get it. And if you get it, you get tens or dozens or hundreds. This is what it looks like up close and personal. Lyceales. The most Lycogula belongs to this group. Tubifera, which you saw as that pseudoethallium, belongs to this group. Cribarius belongs to this group. Awizia we don't have in this part of the world, so forget it. But this is what it looks like. The Lyceales presumably are thought not to have a capolitium, but a paper was published about 3 years ago making a strong case that this member of the Lyceales, those bristles that you see, that's actually a capolitium. That's old Lycogula we've seen before. Ooh, that's Tubifera that we've seen before. This is a little Cribaria, actually very long stalk, quite common on pine wood or on conifers. And this is that Japanese lantern that you saw before. That's pretty intricate. Again, you're talking about something, the whole fruiting body is only a couple millimeters tall. Trichiales, these are the ones with bright spores, yellows, oranges, and things like that. Arceri, Hemitrichia, Betatrichia. We have all of these here. And they have a rather elaborate, most of them have a very elaborate capolitium. They said, whoa, that's really elaborate. Well, it's incredibly so. When you're talking about the diameter of that is 4 or 5 microns. Incredibly intricate. You go to electron microscope, looks even more intricate. How in the world is that gabagoo that was a plasmodium develop into something like this? This is a cute little guy that has long spines on it. You can identify the species just seeing that. It's only one. It's Parakina chrysosperma. That's neither here nor there, and a couple spores. This is actually another Parakina, and you see that the capolitium, the elements are all linked together. In some of the Trichiales, the pieces are separate, and others, they form a nice network. This is a little Trichia, and the only reason I'm showing you this is that sometimes the peridium and the capolitium and spores are the same color, and sometimes they're not. And this one they are. This one, Hemitrichia serpula, ooh, obviously the same color. That Arceri cinerea that you saw earlier, well, same color there too. And this is that one that talks about the long stalker stipe, Hemitrichia caliculator. If you look at the top, you can actually see the individual strands of the capolitium at this magnification. Same genus, Arceria. Some of them are red, some of them are not. They vary. This is a little guy, Trichia Veracosa. If you ever take a trip to Australia or New Zealand and go in the bush, when there's been at least some moisture, this is very, very common. Trichia Veracosa, sometimes large fruiting. I have collected in North America for a very, very long time. It was at least 30 years before I found the first fruiting. It's a weed there and very rare here. Why is that? Again, I don't know. It's interesting. This is Metatrichia vesperium I saw earlier. Kind of cute. This is Parakina Depressor because the fruiting bodies, which are sessile, are depressed. Then the Stevenson, things like Stemonitis, they have a capolitium. There's some capolitium and some spores. And the genus Stemonitis, the capolitium can be rather extensive. And there's actually the very edge of the capolitium forms what we call a surface net. Sort of like looking like chicken wire. This is a common Stemonitis. If you walk in the woods and look around, you're going to see this. It's actually Stemonitis exifera, but you don't have to remember that. Now, this is important. You say, well, that's a Stemonitis. Is it the same one as I just saw? Probably not. What's the difference? The substrate. One of them was on wood. This is on litter. Probably two different species. Things have, I mean, there are Mixomycetes that associate with bark, decorticated wood, litter, and sometimes there's overlap, but seeing that on litter and large fruiting, I'm pretty sure it's different species than one you just saw. This is a little Chamatrica. You can see that it has a peridium, silvery peridium, peels off. This is a Lamproderma. I mean, it looks like tinfoil. The biggest group of Mixomycetes are the Fizzerailies. These have lime. Books will say calcium carbonate in some part of the fruiting body. If you don't know whether you have one of these, you just take one fruiting body, put it on the surface, slide or whatever else, and add a drop of an acid. Acetic acid will do. In other words, vinegar, and it'll bubble. The lime will bubble, bubble, bubble. You know, but you usually can spot it. You don't need that. This is lime showing it. The capolitium has lime in it. And this one, those large lime nodes, I mean, you know that they're lime. There's mounted on a slide. You can see the lime nodes, which can be different shapes, different colors. Ooh, these are really big lime nodes. That's very different looking, but they're lime nodes, again, spores. Oh, wow. Look at those spores. You said, well, I bet I could identify that one. You probably could. Those are very distinctive. Well, there are several species that have spores like that. That actually came from a Lamproderma. Oh, what's this? Well, some spores are not necessarily round. This one actually has ridges on it. And when it's dry, it really collapses along those ridges. Very distinctive. And if you see this, see the spores, it's a physerum. You say, I bet I know what that is. You'd probably be right. Wow. Another reticulate spores, little fences across the spore surface. Wow. I know what that is. Phyllogoseptica. Wait a minute. Do they feed upon rocks? No, they don't feed on rocks. That plasmodium can migrate some distance. How far? We don't know that either. But certainly that plasmodium has migrated, my guess, at least a foot to get up to that high, dry place, which is the best place to be if you're going to disperse your spores. Ooh. Someone sent me this image. I've never seen this myself. Sent me this image some years ago. That's a fresh Phyllogoseptica and a little mammal has walked across it. Those are tracks. I took it to a mammologist. He said, I can't identify it. I don't know what it is. Oh, pretty. This is a tropical species. species, actually tropical genus Fizzerailies. This is the diderma, flat on the surface. Diderma, wait a minute, derma, skin. This one has 2 layers of the peridium. Ooh, diderma, 2 skins. This is a little guy, Fizzerailies verdi. It's just cute. It's actually pretty common in Eastern North America. Ooh, if you have the photographic equipment, when it dehisces, when it opens, it can form that floriform, that flower-like configuration. Again, miniature. You really have to look close to see it, but that's kind of neat. This is a little guy with an orange peridium. It's a Physerailium, a white stalk. It's called Physmelium. This thing is most common on aerial litter. That's where you find it in the tropics. This one, if you have a little bit of imagination, most of you know what clams are, bivalves. This is Physerailium bivalve. I mean, it opens along a suture along the top like a little clam. This is actually another aerial litter Mixomycetes. The peridium on this one, it doesn't really show as well in my images I'd like. It looks like powdered sugar because the lime occurs in crystals. It's like you were powdered sugar on a cookie. This is the one, some of you may have encountered this, certainly a lot of books mention it. In the summer, sometimes you go out on your lawn and see some of the grass that has something white on it. I'm not talking about a fungus. And you look at it and it looks like little bodies. Well, it's probably a Mixomycetes, Physmelium. Well, occurs on litter of this type too, but it apparently lives in the soil and migrates out to fruit. And that one that you've seen before, to me, it's breathtaking. Don't laugh, or if you do laugh, don't turn on your mics. I don't want you to offend me, but it's neat. This is that actual little Physmelium. You see the hypothallus? Ooh, what's this? Well, they're fungi that parasitize Mixomyces. And what you're looking at, he said, well, that's an eraser of pencil. They're awfully small, but this is one guy that actually parasitizes members of the Trichiales. And once it parasitizes, there's not going to be any viable spores. It's dead in the water. Oh, that's a plasmodium. I would hope that most of you already know what a plasmodium looks like, but if you didn't, I should quiz you. What's that? And you'd say plasmodium. I hope you would say that's a plasmodium. This is important. You know this is important. That's a purpose for any club or organization. Most people are ignorant about slime molds. I didn't put the period after ignorant. Okay, they're ignorant about slime molds, but what do? You inform them. This is a workshop held at the University of Arkansas for middle school teachers and their students, learning mixos, how to collect them, how to grow them in moist chamber cultures, and the whole bit. The book that he's using is one of my books. Shouldn't put in a plug, but it's useful for this. Well, that student went to Central America and did some workshops. Wow, all of those people attend a workshop on Mixomycetes? Yep, they did. A lot of interest. Ooh, that's me in the background. This is northern Thailand. I actually gave a 3-day workshop. And I have students from, I got to think for a moment, at least 4 different countries. And they enjoyed the workshop and learned a lot. And in fact, one of them earned a PhD under my direction. Another one visited me as a Fulbright scholar. And the young lady here, the one front row, all the way to the right, she's the one that was growing that plasmodium in that plastic container, looking at biodiesel and whether this thing, Mixomyces, can help fight cancer. So it was neat, I thought. All right, what have we learned? What have I learned? What have other people learned? Mixomyces occur in every type of terrestrial ecosystem examined to date, period. They're not very abundant in some communities. Nothofagus forests are awful. You can collect for an hour and not see a one. Some species are incredibly abundant, surprisingly abundant in deserts, arid regions of the world. And if you keep looking for them in all kinds of new places, what you're going to see happen? Species new to science popping up. I actually had a species that's probably new to science pop up in my front yard, but not exotic. What is there left to do? My career is nearing an end. Talk is nearing an end too, but one will outlast the other one, I hope. Well, examine distance or evidence for long distance dispersal. We don't know how much it occurs. I mean, that example I told you about, southern hemisphere, it's everywhere. Northern hemisphere, it's incredibly rare. Why can't the spores get blown around? I mean, 10 microns, the wind will pick you up. We do need to carry out additional survey work in regions of the world that are still very much understudied. And we need to develop, and we've made a lot of progress, especially over the last 20 years, understanding global patterns of biodiversity in Mixomycetes. You know, where is the highest biodiversity? What are the patterns? What are the trends? And something we're getting into, one of my former postdocs is leading the way on this, direct environmental sampling for Mixomyces. In other words, extract DNA from soil, sequence it, and find out what Mixomyces are there without ever seeing a fruiting body. Wow, that's the future of it. Wow, isn't that a neat place? Not very good for Mixomyces, it's too darn wet. Well, probably not good for Mixomyces, too darn dry. Well, surprisingly, you do find Mixomyces both places. And this is in Namibia, in Southern Africa. And a really interesting thing, there are substrates there. We actually, on this expedition, found several species that were not previously known outside of the deserts of South America. So they obviously hopped the ocean to get here. How, when, we don't know, because this desert is supposed to be the oldest desert in the world. Are they there? Well, in my lab, I'm currently working on material. This is north of Adelaide in the outback of Australia. And you say, well, that doesn't look like a good place for Mixos. You'd be surprised. I love certain kinds of food. I love my wife. She's within hearing distance, so I'm just saying that for her, but she already knows it. I love New Zealand. I've made, I think, 18 trips to New Zealand. I've surveyed the country from top to bottom. Well, and including the subantarctic islands off New Zealand. This is one of the only examples, in fact, it's the only example I know of a discrete pattern of showing latitudinal biodiversity, the pattern. In other words, increasing biodiversity as you go from north to south, from south to north into the southern hemisphere. Those are the numbers of species of Mixo. 25, you get way down south. Number picks up a little more, and you get to mainland New Zealand, and there are quite a few. For the last few years, in fact, 3 years ago, I went to this island. I tried to bring in island biogeography. In other words, you go to an isolated island and you find something there. Obviously, it got there usually through long distance dispersal, but you have to know what's there. Norfolk Island is a neat place to go. And again, my wife was with me, went there. The Island of Pines. Whoa. Wait a minute, Norfolk Island pine. That's where Norfolk Island pine comes from. That's where it evolved. This is another place called Christmas Island. It's in the Indian Ocean. And those are study sites, no big deal. You can see it's off Australia. I went there 4 years ago. And I can tell you, found a species new to science, no big deal. This is a forest on Christmas Island and something that had not really been reported for Mixos before. Those are large land crabs. And what they do, they eat plants and forest for litter. You go into a forest and there's hardly any litter. Well, wait a minute. You're removing a substrate for Mixomyces. So you say, wait a minute, crabs affect Myxomycetes? Yes, they do. Yes, they do. These guys affect Mixomycetes. Well, not hardly. This is in the subantarctic. Those are penguins. That's another one of the islands that I looked at for island biogeography. The Southern Ocean, the subantarctic islands are incredible places. They're kind of raw and hard to get to, but they're unique on this planet. They're really unique on this planet. And yes, they do have mixos. And one of the things I did, just published a paper on it. What are the differences in mixos in Christmas Island in the tropics and Macquarie Island, which is the shot you just saw, in the polar regions? You know what? There's almost no overlap in species. No overlap, or almost none. This is something that attracts people's attention. Art. Oh, wow. If you're good at photography, you can get things like that. One of my students did that. And there, you can see that plasmodium you saw earlier. Or you could paint. Someone I know painted this. This is Trichia Veracosa. And these are, I've seen some really superb illustrations of Mixos photographs and paintings. But, whoa, that's a neat day, isn't it? This is actually in Mongolia. Now, there are rainbows there too. As I tell people, there's an old wives' tale, well, more than just old wives, I guess, that you find something at the end of the rainbow. It's called a pot of gold. That's not true. What you find, what you really find, a pot of Mixomyces. Yes, yes. And I thank you for paying attention to my rambling on.
Thank you very much, Dr. Stevenson. I would say that all those of us that have experienced the fungal world got into it largely because it was so esoteric. So you've opened up a whole new rabbit hole of beyond as an esoterica for many of us. So we probably have a few questions in the chat room. Elizabeth, you want to moderate that?
Yeah, sure. One of the questions was about what the slime molds are eating, and do they focus on the cellulose or the lignin, or is it just more generally all of the plant material?
Well, slime molds are not decomposers.
Okay.
They actually feed on the microorganisms associated with dead
Oh.
organic matter. Now, I say that we now know that a few Mixomyces in the genus Physmelium do produce cellulases. Well, they never touch lignin. But if you go to a rotting log, it's just teeming with bacteria. The soil is loaded with bacteria. That's what they feed upon. But they also feed upon algae, fungal spores, probably little bits of hyphae. That's their diet. That's their main diet.
And at one point you called them critters in passing. Do you think of them as critters?
Yep. That's just a pet phrase of mine. You guys are you guys are critters too. So
Because one of our members asked about how fast they can move, and I think a lot of people have probably seen the studies of, you know, the slime molds that someone got to grow into the shape of
maze,
the maze of the subway system and things like that. And
Yep.
Can you talk a little bit about that?
Well, a couple of Japanese Mixo people did something that anyone could have done 50 years ago, I guess, but they were the first to do it and demonstrated. But now that the rate, you're really talking about no more than at most a few centimeters per hour. They don't race along. If you're going to set something up like that, set it up, and maybe 2 days later it's done what. And of course, the maze cannot be very large. They're limited on how far they can migrate. But no, they don't. Again, they don't zip along. I have known instances in which I saw a plasmodium on a log or in a litter, and I'd go back several hours later, and I'm going to collect the fruiting body because I assume it was going to fruit. And I go back, and the plasmodium is no longer there. So we're talking about at least half a day. But in that period of time, yes, they can migrate back into the litter or migrate back into a crevice in the log. And a plasmodium can go through into a crack, any opening in wood that is large enough to accommodate a single nucleus. Wait a minute, that's pretty small. I mean, they can migrate. I mean, a plasmodium can migrate all throughout decomposing wood. I mean, even through the vessels in the wood. We don't think of that, but they're there and they're doing the thing.
And how long do they typically spend in that plasmodium stage versus the fruiting stage?
Give me an answer.
Hours, days? Is it
We don't know. I mean,
is it varied?
there are some strains of Fizzerailies that you can maintain. I guess they're immortal. I mean, you can maintain them as long as you want. But typical Fizzerailies polycephalum, which is what I'm talking about, if you expose it to light, I mean, the fruiting has to be triggered. Now, some of them, light is the stimulus that does it. In some of the cases, lack of food or whatever else. But once they start fruiting, that's it. They're not going to go back. It's totally determinate. But certainly, well, that amoebo-flagellate stage, some of those may never fruit, so they really are until, I mean, they keep dividing, but they live forever. So, but for common Mixomyces, no one's ever really done a study of that,
Okay.
not to my knowledge.
We did get one comment from a professional mycologist in our club who says she's very excited to have met one of her mycological heroes. So I wanted to pass that along.
Again, I didn't catch the first part of that.
So sorry, one of our club members is a mycologist who works at the USDA, and she said you're one of her mycological heroes, and I just wanted to pass that along.
I do know a couple
you've
people.
been in the
there.
field for a long time, and she first came across your work in New Zealand, and
so can you, what are the slime molds? Are they in the kingdom Protista now, or have they moved that around again?
Well, in supergroup Amoebozoa, but in the earlier classification, Protista, yes.
So it's Amoebozoa is the official kingdom
Yep.
now, right?
Yep.
Okay.
And then one last question, which is one of our members asked if you have any tips for taking good pictures. You have such detailed close-ups of the fruiting bodies, especially.
Have good friends with the equipment. I'm not kidding you. I used to photograph a fair amount, actually before before digital cameras came in. And I got some reasonable images, but it was incredibly time consuming and taking me away from collecting and doing other stuff. So I basically turned it over to a couple of students who got enthused with it. And now I have some colleagues elsewhere in the world. I have a colleague in Russia that he has equipment in his lab. He can do wonderful stuff. I have another colleague in the Ukraine does wonderful stuff. Some of the Japanese do incredible stuff. I mean, it's why spend all the time if they're willing to let me use their images.
Right.
There's no future in the other alternative. I might be lazy, but I do things like edit their papers for English. I make up for it.
Sounds good. Share the work. And Tom was asking about what we know about sort of evolution, and I guess that ties in a little bit to the kingdom question and how long have the slime molds been around compared to other organisms?
Well,
do we know?
They appeared in 1954, believe it or not. No, wait a minute. No. Well, if you use the molecular clock, if I could say it, I mean, it's not absolute, but it gives you some indication. Well, the molecular clock data would indicate this group is probably 1 billion years old.
Wow.
Now, there people find fault with that. So wait a minute, there wouldn't have been a habitat or microhabitat on land for them then. Well, maybe yes, maybe no. Certainly there were bryophytes, algae. I mean, fossil record is still incomplete. I mean, especially that far back in deep time. What I'm saying is there probably were plants, maybe not 1 billion years ago, but maybe 500 or 600 million years ago, certainly by 400 million years ago. So the thing that is thought-provoking, I've already indicated in one of my slides, the Mixomycetes are found everywhere, right? They tend to be abundant if you look for them. Now, anything that's found everywhere and it's abundant, and apparently has a long history, why don't we give them more attention? Why aren't they more significant? Well, they have no economic value other than, well, I don't know it's ever going to be a major source of biofuel, but I mean, possibly some of the compounds they produce might have some value, but for all intents and purposes now, they have no economic value. I said they were not toxic. You could rub a Myxoplasmodium in your eye and you're not going to get any irritate. I mean, they're totally, totally harmless. They're not pathogenic. They'll never give you anything. So with those characteristics, people ignore them. Now, the exciting thing about fungi is that some of them will do bad things. That makes them more interesting. Mixomycetes are so tame. They're, well,
right. And a lot of the paying jobs in mycology are people studying pathogens, right? And so it's, yeah.
Well, they don't study Mixomycetes, do they?
Right, right.
Well, you know, until Alexander Fleming discovered the orange mold and discovered penicillin, nobody cared much about fungi either. So I suspect it's only a matter of time.
Well, we went through a period, well, we're still in it, but 20, 30 years ago, it sort of reached its peak of bioprospecting for active substances produced largely by filamentous fungi. In fact, some of these exotic places I've been, I actually did some collecting for Merck Pharmaceuticals. They paid me a small amount per sample. It was cheaper than sending somebody there, but if one of my samples yielded something that earned them $1 billion, I'd never even hear of it. I got nothing out of it, or would get nothing out of it, but they're still doing that because the biodiversity of fungi on this planet is mind-boggling. We tend to concentrate on the macro guys, but there are a lot of little guys, and they're probably a lot more. Right now, 1,000 the number of formally described species of Mixomycetes is between 1,100 and 1,200. I don't think it's been published, but we are discovering that there are cryptic species. They're biological species which do not correspond to, I mean, a single morpho species is what we recognize. We examine it, and that morpho species is made up of maybe 6 or 8 biological species. They all look alike. Wow, how do you handle that? Well, as an ecologist, I'm not prepared to do that, nor is anyone else. There have been a very few studies that have tried to unravel that, but very limited and very difficult and very expensive because of the molecular work you have to do. So I don't, some of my students have done molecular work, but I'm an old dog and I quit learning new tricks many years ago. Okay.
So Barb's asking, you mentioned that they're eating bacteria among other things. Would that be one possible route of investigation for some useful purpose?
Well, yeah, and most of the investigations are quite old. Some of the classic papers date back to the 1930s. Actually, people feeding Plasmodium different kinds of bacteria. And they found out that, well, if you go to, well, a salad bar and you walk down with a plate, most people are going to pick up a little bit of this, but maybe not that, and move on down and pick up a little of some third item. They're not going to pick up everything. Well, Mixos don't feed on all kinds of bacteria either. They reject some bacteria. They feed upon only certain ones. Now, what the full range is, goodness, there are too many bacteria, but these were simple little studies that were done a long time ago, but it shows that Mixomycetes, and they were using just one species of Plasmodium. You don't know that it applies to all of them, but Mixomycete Plasmodium are discriminating. Wow. They're sophisticated. They have taste. Well, sort of.
Yeah, maybe so. Well, this has been just fascinating, and I feel like
Real quick, I think you may have visited our club some time ago and had some wet culture going and put them in under a dissecting scope, and you had a plasmodium on there. And I still have not forgotten that, and I still talk to people about it, watching the fluid flow back and forth to distribute.
Shuttle streaming, it's called. Shuttle streaming.
Yeah, but anyway, that I'll always remember that. I was kind of hoping I might see a video of it on your presentation.
No, not in this, not in this.
Anyway, but thank you. It was
Just the bare facts.
Yeah, it was very good. I think I've been through Micromycetes 101, so we're all at least at the ground base of attending your future courses. Thank you so much for your time and
I will put a
explanation.
plug for something.
Okay, you have the right to do that, certainly.
Yeah, I'm desperately poor. And no, that's not it. I'm actually teaching a 10-hour course on Mixomycetes. It goes into a lot more depth than this for the Eagle Hill Institute in Maine. It's an online course taught in 5 2-hour sessions in October. If you know of anybody interested in something like that, they can find it on the web. I can't give you a web address, but
If you send something to us, I'm sure that we can put it in the summary notes from this meeting. People might be interested.
well, the only thing is the flyer on it hasn't been developed yet. I don't have it. They have not posted. I'm just telling you that because I know it.
I see.
The director of the station knows about it, but there's just 2 of us at the moment that know anything about it. So, but yeah, I can pass something along, no problem.
Okay, any other questions or comments before we close for tonight?
no, thanks everyone for coming.
It's been a wonderful evening. Thank you, Dr. Stevenson, and thank you, Mitch, and everybody else for a well-spent evening and learning about the wonderful world of fungi and its allies.
Yep.
Or
Okay.
maybe slime molds and their allies in honor of our guest.
Well,
So-
actually, the slime molds are probably older, but although there's some very ancient fungal fossils that have turned up recently.
Yes,
Some
we'll
hyphae.
probably never settle that finally.
No, you can't go back in time,
Right,
unfortunately.
not yet.
Not yet. Well, I don't know. Okay.
Probably not ever. I'll go with that.
Okay.
Okay, so good night, everyone, and see you next month.